Good morning, everybody. I can see we've got a few here today, which is lovely to have you all here. Welcome. My name is Catherine. I think most of you know I'm the CEO of the Baird Institute. We're here today to present on our cardiac regeneration research. The webinar today is going to outline the therapies that we are exploring that could amplify the heart's natural ability to regrow after a heart attack and in time help hopefully reverse heart failure by allowing patients to regenerate their own hearts. Our hope at the mid-year appeal was to raise enough money in order to fund a postdoctoral fellow who's going to be working in this research. And we have Dr. Julia Martina here today, and she'll be speaking a little later. And she is that postdoctoral fellow. So welcome to Julia. First time she's been on one of our webinars. And then today we've got our chair of our institute, Professor Paul Bannon. Doesn't really need much of an introduction. I think most of you on today have met our other key people in the institute. There's Professor Sean Lowell, cardiologist and one of the co-directors of the Centre for Heart Failure within the Baird Institute, the Centre for Heart Failure and Disease of the Aorta. And then we've got Dr. Robert Hume, was also one of our postdoctoral fellows. He is now obviously still part of the team and is now lecturing at Sydney University. And we've got Daria here, who's our tech guy, and myself. So I'll just pass it over. Just a little bit of housekeeping. I just wanted to say all attendees' microphones have been muted. So just to be sure that the webinar goes smoothly. And if you have any questions throughout the presentation, we will give you a bit of time at the end to ask those questions. Just note them down and we can get to those at the end. Just let everybody speak first and then everyone will be here to answer your questions. So I'll pass over to you, Paul.
Thanks, Catherine. Welcome, everybody. Look, I think this is a very exciting session to have today. It represents, we've got three superstars here with Sean Lowell, Rob Hume and Julia. Sean has been instrumental over many years and in developing what we call the Biobank or City Heart Bank, which has a sort of a joining of forces of the aortic bank. So many patients that have had aortic surgery, we try to get hold of that tissue for research purposes, and he's joined that with the cardiac bank. So, and this is an incredibly successful program that's allowed an enormous amount of translational research over the years and much more to come. We've focused in the past about some of the, and in fact, the symposium coming up soon has got a lot of work on research coming on the aortic side as well as the cardiac side. But today we want to talk about the cardiac side in particular. And of course, there's been some exciting research recently published as well. And I think it'd be great to hear from all three of them. So you've got Sean from cardiology side and overseeing Sydney Heart Bank. You've got Rob, as Catherine's already spoken about, remains as head of translational research for the Baird Institute, and of course Julia, who's doing probably most of the work, I imagine. So that's the way it pans out, of course. So I'm very keen to hear it all. Although I primarily focus on aortic surgery, I'm really keen to hear the strides that we've made on the cardiac side. So I'll end there and hand to Sean.
Great, thank you. And I will share my screen. Okay. Can everyone see that?
Yes.
Great. Thank you very much, Paul. So I will start things off just briefly to give some background regarding the cardiac regeneration program that is heavily supported by the Baird and donors such as yourself, and we're very grateful for that support. And we'll talk about that program and give you some background. I'll give you some background as to how this program came about. And then I'll hand over to Rob, who will talk about the exciting publication that came out earlier this year that was a world first in really showing that the human heart can regenerate. So we'll go from there. And then Julia will finish to actually talk about the work that she will do to carry on this program, where we can really build upon the findings that we have and hopefully translate that into therapeutic options to regenerate the heart and reverse heart failure. So I don't need to tell the audience here that cardiovascular disease is a challenging and increasingly important public health measure. And for this reason of why it affects so many Australians, and because there are so many unanswered questions, that was really part of the basis of why we formed the Centre for Aortic Diseases and Heart Failure. Now, something that underpins a lot of the research that we do is human heart samples, because animal models are very important. They're preclinical models, but obviously they're not the human heart. And so we're lucky enough to partner with the Sydney Heart Bank, for which I'm the director, and look at the heart samples that have been there historically. And then, of course, as Paul mentioned, there are numerous collections now that come from RPA, led by Paul, and that's why Paul leads this with me as well. But, it's important to acknowledge the history there of the heart bank. You can see it began in 1989 by my predecessor, Christos Remedios, who's now an emeritus professor, but he was my undergraduate honours supervisor, and that's how I really became involved. And of course, the late Dr. Victor Chang from St. Vincent's Hospital. And as such, it is the oldest and largest human heart bank in the world, and has been supported by the University of Sydney and the Baird Institute more recently, and it partners with both St. Vincent's Hospital in Sydney with its historic samples and Royal Prince Alfred Hospital. So as you can see here, it's been housed in at the University of Sydney in the Charles Perkins Centre, and it has been supported by the University of Sydney since its inception in 1989. And hence there are these historic samples which have been collected from patients who have had a heart transplant at St. Vincent's Hospital in Sydney. And then there have been more recently, and this is where the partnership with the cardiothoracic department at Royal Prince Alfred Hospital, and principally Professor Bannon, where there is now myomectomy tissue and samples taken from patients undergoing bypass and valve surgery, of which we'll talk about today. We've partnered also with the vascular surgeons to collect vascular tissue, so tissue from other blood vessels. And of course, the aortic bank, which we're not talking about here, but which Paul started many years ago, has merged with the Sydney Heart Bank. And so there's a unique collection of aortic tissue as well, which also forms the basis for the research that is undertaken as well. But the main point being is, the Sydney Heart Bank is a unique resource. It's over 40,000 cardiovascular samples, and I guess the name probably has to change at some point from being a Sydney Heart Bank to a Sydney Cardiovascular Bank, because of those aortic tissues as well as the vascular tissues. So just about cardiac regeneration, it's really fascinating when you think about the history of cardiac regeneration, because it began with these animals such as the zebrafish, for example, where there was an observation that if you actually cut the tip of the heart off the zebrafish, it completely regrows, which is an amazing concept to get your head around. And there are other animals as well, like the salamander, that can do the same. But obviously, a zebrafish is nowhere near a human, but it's also not a mammal. So the next lot of animals, I guess, that were looked at were rodents, and in particular when they looked at mice. And mice obviously being mammals, but not being humans, of course, it was an important first step. And interestingly, it wasn't the same case that you could take an adult like you could with the zebrafish, cut the tip of the heart off, and it would regrow. That wouldn't happen in the mouse. However, there was this window of opportunity just after birth, where indeed, if you caused an injury to the heart or cut the heart tip off, there was an ability for that heart to regrow, but then it was essentially lost in adulthood. But it was exciting because the mouse is a mammal, and then we thought, well, could this also translate to humans? But there are some problems when you are trying to relate data and studies from rodents to humans, even at the very basic level. If you look at a heart cell, whilst in the neonate, both in a human and a rodent, they look similar. They're a cell that has, for example, a particular nucleus with a certain content of DNA, and there's one nucleus, as you can see here, in the middle of the cell. But then the problem becomes between the adult heart cells, between a human and a rodent, there are some differences. The rodents can have two nuclei, for example. And so there are some inherent things that we have to keep in mind. Now, when we're trying to study cardiac regeneration, what we're looking for is evidence of a heart cell increasing the amount of DNA in its nucleus and then essentially dividing to form two daughter cells. So essentially you're having one cell, which is an adult cell, become two cells, because that is the very definition of replication or regeneration. You're forming new heart cells from the existing cells. And the textbooks will tell you that the human heart is unable to do that. But there were some landmark studies, and one such is from Harvard Medical School, and this was back in 2013, where they did something very clever to do with a mouse model where they looked at heart attacks. And so what they did was they labeled all the heart cells in these mice, and then they induced a heart attack. And they were able to follow these cells because they had labeled them. And what they showed essentially was that after a heart attack, there were numerous heart cells that were fully formed adult cells that actually divided to form new cells in the area around the heart attack. And when you compared that to a group of aging mice, it was a very low level of new heart cells that were being turned over. And so I was fortunate enough then to go do my postdoctoral training in members of these labs, Claude Lechen and Matt Steinhauser at Harvard. Sam Senyo was still a postdoc, whom I became friends with, and Rich Lee, who was a senior professor at Harvard. And I looked at this model, and I then wanted to see how we could translate this to humans. And I ended up studying human fetal hearts, which you can see here, and culturing them and looking under the microscope to see how they regenerate. And what was extraordinary in these human hearts, in these human fetal hearts, was there was a very high level of regeneration. Approximately about 20% of heart cells that were forming and growing were actually being regenerated. And so that was, a unique finding. And what we did was a collaboration between Harvard and MIT. We used a machine called multi-isotope imaging mass spectrometry, where I actually labeled and this is what I'm showing here, we labeled these heart cells with a tracer, and then using this machine, which essentially bombards the samples, as you can see, there's one sample in there with cesium ions. It's almost like a nuclear production. And what it does is it obliterates the tissue, and then you capture the ions that are emitted. And because you labeled the cells, you could actually see human heart cells taking up this label from the culture. So what it essentially allowed us to do was look at these human fetal hearts and then see if they were capable of regenerating, because the tracer that we used was a DNA building block. So if these heart cells were taking up this DNA building block, it was because they were trying to make new heart cells. And what we essentially showed was somewhere of the order between 16 to 20% of human heart cells in the fetus were actually being made anew. And so that was quite a novel concept. But if we go back to this model of a heart attack, what I wanted to know was, in adults, is there a possibility that you can make new heart cells after a heart attack, similar to what they showed in this mouse model? And now knowing that we can see that a normal human heart, as it grows, is trying to make heart cells, the next step was trying to see, after a heart attack in a human, can you also make new heart cells? Because that had never been shown before. So with Professor Paul Bannon, we devised a way that patients undergoing heart surgery, bypass surgery, after having had a heart attack. So this is the important point. These are patients who have come into Royal Prince Alfred Hospital with a heart attack. And then when the angiogram is done, they have numerous narrowing stenoses in their arteries, which mean a stent is not the best option for them in order for their heart to recover, and bypass surgery is the best option. And so they remain as an inpatient. And it's during that time that we expect there to be some growth, well, the hypothesis was at least similar to this mouse model, that there may be some regeneration that happens. And so our proof of concept study, and this is novel, and it's still a novel biopsy program around the world, is Professor Bannon biopsied an area around the infarct, and I would look at what area, that was based on the angiogram, and we even did ultrasound, a transesophageal echo during the operation, and we looked under direct visualization and biopsy the area around the infarct, and then also took an area away from the infarct. So you had a control. And what was extraordinary in our very preliminary studies was, sure enough, around the area of the infarct, similar to that mouse model I showed you, but now in a human, you could see human cardiomyocytes, as evidenced by these red dots, which are a marker of turnover of heart cells, were actually increased in that area around the heart attack compared to the normal area. And so this really set the stage then for us to undertake a world first study. And this is then when Rob came on board as a postdoctoral fellow at the Baird Institute. And he did some amazing work to take these preliminary results and observations. And as you can see, it involved a great team of people because it was such a landmark study, and it was published in January of this year. And I'll leave it now to Rob to take it from here and talk about that study.
Thanks a lot, Sean. I will just share my screen. Okay, can everybody see that okay?
Yep.
Yep. Okay, great. Awesome. What an absolutely brilliant introduction. Done a lot of the heavy lifting for me there. Thank you very much, Sean. That's really, really great. So yeah, let's see if I can progress this slide. Doesn't seem to be. Oh, there we go. Didn't realize that was going to have sound on it. Okay, sorry about this. There we go. Hopefully you can see these videos. Hopefully they're projecting okay. Yes, the work that I do specifically is on this cardiac regeneration work, and also on this vascular regeneration work as well, where we make these degradable grafts and we put them into arteries, and then they degrade and remodel over time to make these nice new arteries. So this is kind of one arm of my research, and the other arm is the cardiac regeneration stuff. And today we're going to be focusing on this cardiac regeneration work. So as Sean has already introduced you, one of the main things that's going to be kind of the theme of this talk, and the main the star of the show, is the cardiomyocyte. So what's a cardiomyocyte? Well, it's the heart muscle cell. The heart's full of lots of different cells. You can see it's full of blood vessels. It's got cells that produce collagen to keep it structurally sound. And it also has these cardiomyocytes. And what are the cardiomyocytes? Well, they're the cells that actually contract and really work in unison with that electrical impulse to pump blood out of the ventricles and through the body to your organs, to provide blood and oxygen to your organs. So Sean's already covered this very, very nicely. So we know from what Sean just told us that cardiomyocytes, the question used to be, can they actually undergo cell replication, or what's known as mitosis? So can one cell divide into two cells? That was a big question for a long time, and lots of people thought the answer was no. And because people thought the answer was no, then they thought, unlike other organs, it was previously thought that the heart really can't undergo regeneration because these cardiomyocytes can't really undergo this mitosis. So the kind of question used to be, are the cardiomyocytes you're born with the same cells that you die with? And basically, in scientific terms, we're asking, can these cardiomyocytes undergo mitosis? So Sean's already told you some pretty cool studies that led to this, but I just want to tell you about one more that I really, really like, just because it's a nice kind of left field way of thinking of these things, and a very, interesting, different idea that these researchers had back in 2009. And what they discovered was that during the Cold War, there was lots of nuclear bomb testing. And you're probably thinking, what's this got to do with hearts? This doesn't have anything to do with hearts. But what happened is during this nuclear bomb testing, the atmosphere became full with carbon-14. And that carbon-14 then became CO2 with carbon-14, so carbon dioxide. Then plants took up that carbon-14, animals ate those plants, or humans ate those plants, or humans ate the animals that ate those plants. And in one way or another, humans consumed carbon-14. And that carbon-14 kind of labeled the hearts with carbon-14. So it labeled all your cells with carbon-14, including your heart muscle cells or the cardiomyocytes. And with this, they were actually able to, in a way, you can think of it as like carbon dating your cardiomyocytes. And if they split them, they would have less carbon-14. And in this way, they were able to understand how many times they divided. And they were actually able to show that cardiomyocytes do replicate just at incredibly low levels, about 0.5%. I think this has been updated to about 0.1% renewal per year. So what happens when you have a heart attack? Well, when you have a heart attack, what's otherwise known as myocardial infarction, you have these vessels on your heart. They're called the coronary vessels, coronary arteries, and they feed blood to your muscle, to the cardiomyocytes. If you don't get blood to the cardiomyocytes themselves, then they don't have the oxygen and energy to actually contract. But when you have coronary artery disease, so a disease of the blood vessels of the heart, you can have this buildup of plaque in there, and then essentially a clot can form, and that might dislodge further down, or it might dislodge further up. The further up, the worse off you are. The further down, the less worse off you are, because it affects less tissue. But what that means is that artery is blocked, and if that artery is blocked somewhere, well, that means there's not oxygen. And if there's not oxygen, then this area below this blockage is all going to start dying, because your cardiomyocytes, they need the oxygen in order to survive and contract. So the consequence of that is all these cardiomyocytes start dying, and that leads to heart failure. So let's just look at that a little bit more zoomed in. So we've got our heart muscle down here, and we've got all the cardiomyocytes in here. And what happens first, they die. So they don't have oxygen, and the area that's affected causes them all to start dying. All your immune cells come in and they start munching up the dead tissue and removing all that dead tissue. And then your body works to replace those cardiomyocytes with a stiff collagen scar. So you just get lots and lots of collagen. Imagine kind of similar to when you cut yourself and the cut is too large, you get that scar tissue that forms that's got lots of collagen in it. So just quickly, let's just have a look at what that means for the actual contraction of your heart. If you tipped your heart over on its side, and then we looked at an image like this is actually a rat heart. It's not a human heart, which enables us to get this full image that you can see here. And then we kind of slice through the heart in terms of imaging wise. This is what we're looking at. So we're looking at one of the chambers. It's called the left ventricle. It's the main ventricle that pumps the blood to the majority of the blood's organs. And up here you can see the aorta. So that's the main blood vessel that it pumps it up to, which then reaches the rest of the body. And what you can hopefully appreciate in this nice, healthy, happy heart is you've got this nice thick wall, and it's contracting really nicely in unison with one another. Over here, you can see this one's had an infarct, and you might be able to make it out, but the trace is kind of going along around here and coming back around here. You might be able to see that these walls over here and over here are contracting nicely, but because the blockage is somewhere around here, everything around here has all died, and it's been replaced by a collagen scar, and it's also become thinner. If I leave my laser pointer just over here, you can see that wall is really not moving at all. It's trying, but it's just not contracting because there's just no cardiomyocytes there to contract. And without the cardiomyocytes, then there's really no muscle. So what I'm trying to get home here is that after you have a heart attack, then the amount of contraction, the actual ventricle is able to contract and pump that blood goes down, and you have less heart contraction. And eventually, this scar can keep remodeling and keep growing to eventually cause heart failure and therefore death. So the question that we were asking, that we've set up in quite a lot of detail here, I guess, is, yes, you've got this big scar forming, but do any of these cardiomyocytes, do any of them replicate, undergo mitosis to form new cells to try and replace that tissue with new tissue? And as Sean explained before, yes is the answer in rodent studies. We know that there's plenty of rodent studies that have shown there is this increase in cardiomyocyte mitosis, but it's never been shown in humans. Important to note that the increase that was shown in rodent studies, very, very small amount that it goes up. So it doesn't go back up to the levels that you see in the fetus, but there is that small increase. So we wanted to see, does that happen? So after you have a heart attack, do your cardiomyocytes, your heart muscle cells, do they undergo cell replication, aka mitosis, which we would hope would cause cardiac regeneration, the return to function for your heart? So I will go through this quite quickly, but there was a super interesting, unique heart that we were very kindly donated by the next of kin, where someone had a full coronary artery blockage, and they were kept on life support for 5 days, and then were very sadly declared brain dead at that point. And the next of kin very kindly agreed to organ donation, and so we were able to collect this heart within 15 minutes. And when we collected it, we were able to collect some of this left ventricle that was infarcted, so that's the LV over here, some right ventricle which had a bit of infarct in it, and some right atrium, which was a nice piece of control tissue. And this is just a bit of histology here where you can see that scars, the scars in red, and the cardiomyocytes are in this kind of orangey color, and you can see this large infarct that is formed in this tissue. So here comes the crux of what we were trying to show and look at. We wanted to know, do these cardiomyocytes go through mitosis, and is that mitosis increased? And so what does mitosis look like? Well, it's this process of cell division. So as you go through these different stages, the DNA, which is kind of here in these squiggly lines, starts dividing and going to either side of the cell, separating, and then eventually you get these two new daughter cells. So we needed to show cardiomyocytes in these different stages, and that's just what we did. So we showed them in the early stages, the kind of middle stages, and even when they're getting towards this final stages, we even saw them actually separating out into two new cells. There's this really, really nice 3D projection here where you can see two nuclei from one of these cardiomyocytes. So the DNA actually splitting in two. But this is all well and good. Yes, we found them going through mitosis, but is it increased? That's the question we wanted to answer. And so, as I explained before, we had this healthy tissue from the right atrium, some kind of partly scarred tissue in the right ventricle, and then the one that has lots of scar and more cell death in it in the left ventricle. And then we were also able to use these really nice donor hearts that were age and sex matched from the Sydney Heart Bank that have been collected and stored in liquid nitrogen for decades and decades at this point. But the tissue remains in incredibly high quality because it's kept in very, very cold conditions. And you don't really need to look at all these different numbers here, but what you can see is these bars going up. And as these bars going up, this is the amount of kind of ischemia or scar tissue that we're seeing in each of these samples. So we've got the healthy donors, and we got the healthy right atrium, and then we got the partly scarred right ventricle and the very scarred left ventricle. And what you can see is the cardiomyocytes increase in their levels of mitosis. These are mitosis markers here, here, and here. They increase in their numbers of mitosis depending on how much of an infarct there is. So what this really shows is that this increase in infarction or increase in tissue damage from this infarction cause an increase in cardiomyocyte mitosis. So this is kind of telling us that when you have a myocardial infarct, when you have a heart attack, your body is trying to regenerate, and the regenerative capacity goes up. I'll quickly go over this. I don't want it to confuse you too much, but essentially we wanted to validate this further than our nice heart, and even in those those heart bypass biopsies that Sean talked about earlier, we wanted to validate it further in a nice independent data set. And that's what we did here. We found this lovely data set online and worked with these very, very impressive bioinformaticians and researchers up at UQ, Nathan Powell-Plant and Woo-Jun Shim. And we looked at this data set, and then this data set essentially has each individual cell has been sequenced, and we've got this information telling us the amount of protein they're trying to make based on their gene expression or their mRNA. So what you really need to know here is each dot represents a different cell. Over here, we have the cardiomyocytes. They're the ones that we're interested in. And when we look at the donor cardiomyocytes, the ones that are undergoing mitosis, and the infarct cardiomyocytes that are undergoing mitosis, you can see each red dot represents a cell trying to go through mitosis. And you can see there's more red dots here, less red dots here. And essentially that just shows exactly what we had already shown in our data. When you have an infarct, so this is infarcted tissue, this person's had a heart attack, or these samples have had heart attacks, and these samples have not had heart attacks, there's an increase in cardiomyocyte mitosis. So those cardiomyocytes are trying to replicate if you have an infarct versus not. So in short, this really validated exactly what we had already seen. So that's kind of just some data from the study, and it's a huge collaboration with so many people, so much historic work done by Sean and Paul before I even started, and lots of other people, and then really important collaborations with people from all over Australia and afar. So way too many people to thank. But it's a huge collaboration with lots and lots of people. So before I move on, I just wanted to get to just a couple of just other points before I hand over to Julia. And the points I wanted to really make is that the only current cure for heart failure, so you can have treatments for heart failure to slow the worsening effects of it, but the only cure is a full heart transplant. And with approximately 140,000 heart failure patients in Australia, and only 115 heart transplants per year, there's a huge disparity between what is needed and what's actually able to be done. So this kind of brings me to my point about the study. And although it's incredibly interesting from a scientific standpoint, you might be thinking these points. So we've got cardiomyocyte mitosis increasing after a heart attack, aka myocardial infarction, but the heart still fails anyway. So these heart muscle cells, they're replicating, but the heart's still failing. So if you've got more cardiomyocyte mitosis, more heart muscle being formed, why are we not getting the heart returning to normal? Why is it not regenerating? And the long and short of it is the amount of mitosis, although it increases, it's still far too low, the threshold needed to actually regrow that tissue and repair the heart. So you might be thinking from a patient's perspective, who cares? Like, that's really cool for you guys to have found that. And scientifically, it's interesting, but what's that going to do for me and my heart? And what are we actually going to do about it? And this is where Julia is going to explain from this discovery what we are actually going to do with this discovery to actually find or try to discover new treatments for this heart failure. So I will pass over to Julia. I just want to mention there's lots of different people working on this work. This is just a snapshot of a few people in the lab, but it's a huge collaboration with people over at the RPA, people at UCID, people all over the world. that we collaborate with, and to get this work done just requires a ginormous team of people. So, and sorry for just very, very quickly, I also wanted to say thank you to you all, because the money that was provided to allow me to do my postdoctoral work in the lab has allowed me to progress in my career, and allowed me to get this lectureship at the University of Sydney. And I just wanted to say thank you for that opportunity to all the donors and all the people that support us. Brilliant. I will now pass over to Julia, who's going to explain where we're going next.
Good afternoon. Thank you very much to Sean and Rob for such nice introduction. And currently I'm going to show you a small window to our lab and how exactly we are going to understand heart failure better, the myocardial infarction better, and what exactly we're going to do to find the new ways to regenerate the broken heart. Can you see my slides?
Yes, yes, we can.
Thank you very much. So our lab has two different avenues. The first one is a disease modeling. So using different models, predominantly stem cell models, we can understand the heart failure better, we can map the heart attack better. And the second one, which is probably one of the most practically exciting, is a drug screening. So we can test different compounds and find the one which can regenerate damaged hearts. So, and for, as I have already mentioned, for these ones, we use stem cells. What is the stem cells and how we get them? So first, we take the blood from the consenting adults, both male and female, and using Yamanaka factors, a Nobel Prize winning discovery from Japanese scientist Shinya Yamanaka, we sort of push a rewind button on the cells, and we restore them to the initial, the earliest stage they can be in, the stem cell. The stem cell, they have absolutely uncurbed potential, and they hold a blueprint to generate pretty much every cell in our bodies. And we're using them to generate 2D cardiac myocytes. So you can see it's a monolayer, single sheet of heart muscle cells, which we can put into the well of the scientific plate as small as 5 squared millimeters. And besides 2D cardiac myocytes, we also can do 3D cardiac organoids, sort of mini hearts in the well. And each of those hearts is just 3 millimeters. Why we try to do as small as possible, to be able to screen, to be able to test as the most conditions as possible, and to cover most of the grounds in terms of the drug development. Also, I tell here 2D cardiac myocytes, 3D cardiac organoids, but cardiac myocytes, heart muscle cells are in the focus here, but we also have different cell types such as fibroblasts, which are making the scar, immune cells, which are, how Rob told, munching the dead cells. So we can definitely target our approaches to the heart cells only, and also look how our different manipulations and intervention influence not just cardiac myocytes, but their counterparts. So Rob started his fantastic research with this unique heart, and this heart has brought a really groundbreaking discoveries to the field. However, this heart showing you a snapshot of many complex processes which happened from the occlusion of the artery, from the heart attack, through the 5 days on the life support until we were able to see what's going on. And this snapshot involves many signaling events, changes in different genes, changes in different proteins. How can we untangle what happened in such a complex situations? And to bypass the requirement for for human heart tissue, we can use stem cells. First, we can literally restore the sequence of events which happened during this myocardial infarction at the time scale. We can compare how the different parts of the heart reacted on this ischemia, right ventricle, left ventricle. We also can look into the atrias. And because our 3D models and our 2D models have different cells, we can literally look what happened if we take one into the equation. So literally look how different cells contribute to the certain damage, to the certain response, and actually to increasing of the mitosis. And after this, we can combine this absolutely unique approach to the most comprehensive portrait of myocardial infarction and heart failure, looking through the genes, how they change the proteins, how the proteins change the metabolites, how they both change the signaling events, and also uncover new biomarkers which we can use to diagnose and stratify the therapeutic options. So I gave you a little bit of an overview to the disease modeling, and I also mentioned the drug screening. So with this drug screening approaches, we can screen like hundreds of the compounds, even thousands. But how we get from those enormous amount of different potential drugs to 2, 3, maybe one specific one, which will be the one. So we start from our 2D cardiac myocytes, then we proceed to our 3D cardiac organoids, and that comprises our in vitro part of the research. And this is a part of the research which prioritizes most types of the drugs, and we are left with a little bit of those which definitely can produce the interesting and desired result. Then we go to a specific model of living beating heart slices. We get hearts from sheep, from pig, and sometimes we get hearts from humans, which were rejected from the transplantation. And we can slice them and maintain their viability in the specific biomimetic chambers. So this is a chamber. When we put the slice between different electrodes, we can pulse them and maintain their viability for up to a month, and so we can test different drugs. And after the drug proved their efficiency in this model, we go to the unique model brought by Sean and Paul, the model of the human myocardial infarction biopsy. So we can finally figure it out which of those potential candidates becoming the lead candidate. And in the end, we can take our failing heart, we can bring the drug and restore the heart function. As Catherine told in the beginning of the webinar, there was a big call to fund my postdoc, and I'm very grateful for you for your donations, so I would be able to join the team. So I would like to tell you about my experience, and I was always a heart person, as I started as a cardiologist in Moscow, and caring for my patients sort of spurred on my interest in the science and how can we do it better. So I started doing my master's in immunology between Moscow and St. Louis in the USA, where I was investigating how we can curb inflammation during myocardial infarction and limit the damage those immune cells bring, which then prompted me to do my PhD in Australia, where I started using stem cells. And I was fortunate to identify a novel mechanism driving human heart muscle cell proliferation, mitosis, which we could harness to develop new approaches for cardiac regenerative therapies. And as Rob had said, and I would like to join, nothing of this research would be possible without the kind donations from the tissue and philanthropic donors and their families. And I'm very fortunate and I'm very grateful to be trusted with these cherished samples so we can bring the most to the science and find something to repair the broken hearts. Thank you very much.
Thank you so much, Julia. That was really all very fascinating, everyone's presentations today. Thank you so much. I'm just wondering, is there anybody that has a question? You can click on the little hand to raise your hand if you have one, and then I can unmute you. There's a little hand at the top of the screen if you had a question. But understand if there's no questions to be had. Must mean that it was all very clear. It all made total sense. Oh, hold on a minute. There's a hand. Ron, let me just unmute you. Okay, Ron, I think your microphone, no, hold on. Can you hear, can you talk, Ron? No, hold on. Should be working.
I think maybe he just needs to unmute himself.
Okay.
Ron, if you look at the top of your screen where it says mic, it'll have a little cross through it. If you try clicking that, it should.
Righto. Can you hear me now?
Yes.
Yeah,
Yes,
perfect.
perfect. Thank you for that, Rob.
No, thank you very much. I've got as much, well, probably more a comment than a question. And if I've missed something, my computer cut out part way through Professor Hume's presentation, but I'm actually an ex-patient of Professor Bannon. And I presented in early 2022 to my cardiologist with breathlessness. I never had a clinically discernible heart attack from external assessment, but he did an echocardiogram and I had very clear damage of my heart muscle. And subsequent angiogram revealed extreme blockage of all three major vessels to the heart. And in October 2022, Professor Bannon performed a triple bypass for me. And I'll take this opportunity to thank him. There aren't really enough words to thank him for saving my life, but there's certainly a lot of thanks for improving the quality of my life back to where I would have been 4 decades earlier. So I'm forever grateful for that. But what I want to reveal is that in a recent review by my cardiologist 3 years after the operation, she said that there is complete regeneration of that damaged heart muscle. And so listening to this presentation is quite fascinating. I mean, I didn't have a frank heart attack. Maybe I didn't have much scar tissue. Circulation was restored, but she said the evidence of initial damage to the muscle was completely gone and the heart muscle regenerated. So it's just mind-blowing how these things happen. And I'd obviously dodged a cannonball, in my clinical history, but it's fascinating, and I just want to congratulate you all on amazing research and thank everybody for the great work they're doing. And Prof. Bannon, again, thanks so much for looking after me back then.
No, Ron. I can make a little bit of a comment about that. I think it highlights a really important point, because you had critical lesions in all 3 territories of the heart, and your heart was in a sort of a dormant state of what we call ischemia. So it was basically in a spot where it was really only getting enough blood and enough oxygen to maintain viability of the heart cells, but your heart function was, but not enough to really perform at its best. So it was a very close call and a good pickup. And if we then perform the bypass grafts in the best way we can, and then add to that the optimal medical therapy, there's a lot we can be gained for, but in returning the heart to good function. But primarily, this was a lucky save. So
glad for that. Yeah.
It is, and because we're able to restore the heart function by getting the blood back to it. But the research that these guys are doing is when people aren't as lucky as that. The heart attack's already occurred. The damage is permanent, unless we come up with something else that can treat that. And I think that's fundamental. And there's a lot of people that do that. And if you think about all the patients, you weren't aware. If you think about the patients with diabetes who have no idea, there's a lot of patients out there that have no idea they've had heart attacks, or so-called silent heart attacks. Yeah. So if there's any way we can reverse damage if we've missed it, if we've missed the diagnosis, if we've missed the heart attack, if there's any way we can reverse that damage, then that's what, that'd be a huge benefit and take the toll off all those heart failure. symptoms, etc. And that's so it's a really good point you make, because you got lucky. That's basically it. And we'd rather not be lucky, and that's what the research is about.
Yeah. Well, nice to have a lottery win, and more power to you all.
Totally.
Thank you for everything.
Totally. I agree.
Any
Yeah,
other
and,
questions?
oh,
Oh, sorry.
I was
Sean,
gonna
you
say,
go.
yeah, Ron, I was just going to say, just to add to that, I think, yeah, it's fascinating, the heart, that it can go into this so-called hibernating state, which is what Paul was alluding to, where it's alive but sort of teetering on the edge, and that's really where, when you can restore blood flow, you've got this window of opportunity to improve the heart function. We can't discount that, perhaps at a cellular level, there might be some regenerative capacity there, and that's certainly an area of research for us, that we plan to look at, that sort of situation where the heart cells are literally teetering on the edge of death. And maybe that is a potential opportunity where, if you could implement regenerative strategies as part of the medical therapy, but also, of course, restore blood flow ultimately. But there would be a window there that you may be able to augment that intrinsic ability to make new heart cells. And that's really where this research has the potential to expand to, not only from the heart attack setting, but to those patients who have some degree of heart failure, because the heart cells are on a stage of where they're potentially dying, and whether then you could actually stimulate them to make new heart cells. So there's definitely an opportunity to expand to that as well.
Yeah, if I can just ask one other question. I'm just amazed how I was able to go for so long, because my initial clinical finding from the angiogram was 100%, 100%, and 90% blockage of the three major vessels. So I must have had some amazing collateral microcirculation just keeping me going. And I was working hard all the way through. I was steaming up the hill to the train station to get to Strathfield Private to get to my operation. I was running up the hill, and I never had any trouble. So I'm just amazed how that situation can occur. But anyway.
So that is
well
definitely
eventually.
some good luck. Forming collaterals is something that we see. We don't fully understand why some individuals do and others don't. There's probably something to do with genetics. There's some evidence around people being very well conditioned. So people who have exercised from their youth at high levels, then you tend to form better blood vessel formation. But you were extremely lucky that, in those arteries slowly blocking off and even becoming completely blocked, that at the same time you were forming collaterals that were keeping the muscle alive. But it was just keeping the muscle alive, and it was probably hibernating. And then when you had the bypass surgery with Professor Bannon, then blood flow was completely restored, and that's why your heart muscle picked up. But it would be a very interesting area to look at, and I do wonder whether a small part of that, there may be some new heart cells that are forming as well, because very similar, not on the same scale as a heart attack, but the biology is very similar, and we could certainly, this is where we would want to expand and look at that. So it's a good point.
But you're not
Thank you very much.
taking Ron's heart muscle, though, Sean.
Yeah, look, I'd love to give you a sample, but best not, if that's okay.
I don't think
with
he's
you.
going to do that now.
No, but I think you could. No, but I think when the situation where you have and like, obviously what we've presented with this model is a unique situation of patients coming in with a heart attack to hospital, and they have to remain in hospital and have bypass surgery, and we have this program to biopsy from around the heart attack and elsewhere. There's no reason why that couldn't potentially be expanded to patients who require surgery in a situation where they have a hibernating myocardium, and we could potentially biopsy that. So that would be another very good area to look at, because it would certainly expand. And I think I'll speak to Paul. We'll speak offline and think about how we could expand that.
It's fascinating. Fascinating. Yeah.
Peter.
Very good. Anybody else have any questions or?
Yeah. Peter,
Peter, yeah, have you unmuted yourself? Up the top right, there, up on the top side, you can click on the microphone. It will unmute yourself. Can you see the little mic? Oh, there you go. I think you're right. No, it's still not. Let me see if I can do it. I don't know that I can. It should be.
Yeah.
Can you talk now, Peter?
I
Yeah.
think you just muted him. Oh, there. He should be able to do it now. Peter, if you just click now in the top right where it says mic.
Right next to there it is. Up next to the camera. It says camera, mic, and then share. Along the bar at the top.
How's that?
That's it.
It was
Can
down
hear
the
you now.
bottom. It was down the
Ah,
bottom, actually.
Sorry, it's on the top for me.
Yeah, Peter Lozanne. Just as a background, I had a double cabbage with Matt Bayfield 24 years ago, and as of my last review, both grafts are apparently working extremely well. So I'm pretty grateful for that at 82 years of age. But as you probably know, my background interest in your research relates to HOCM cardiomyopathy, and from a family point of view. So I'm just interested in how this research might apply to the regeneration of the heart in a HOCM situation. Did we get that?
Yes.
Yes. Just
Yeah.
wondering if
Coming
you're going
in.
to answer, Sean. You want to try?
So are we specifically asking to do with hypertrophic cardiomyopathy?
Yeah.
Is that what
Yes,
you meant
HOCM.
with regeneration?
Yeah.
Well, we didn't go into the background in it, but probably a driver for this capacity to regenerate and why we see more regeneration around the area of the heart attack compared to the area that is normal is our hypothesis is that the lack of oxygen, the reduced oxygen, may be stimulating the heart cells to want to produce more heart cells. And that really goes back to sort of what I observed in those fetal hearts, because in utero, the oxygen level is quite low. So that's the thing that struck me when I was looking at those fetal hearts in Boston, was that you were getting these extraordinary levels of regenerating heart cells in an environment in utero where the oxygen level is 80%. So the heart was able to regenerate. So that was part of the theory of when I came back to Australia to think, well, what's another situation where you have low oxygen levels in an adult heart? And that's really with a heart attack. So we are, as part of the broader program, looking at other types of heart failure because we've got access with the heart bank to look at all types of heart failure, including hypertrophic cardiomyopathy, as well as other forms of heart failure that don't involve ischemia or blocked arteries or a lack of oxygen. And we will look at that. My feeling is that the regenerative levels will be low because you don't have that low oxygen stimulus. But with Julia's work, we may well find ways to, aside from that unique situation to do with a heart attack, find ways that heart cells can be stimulated to make new heart cells. And then, of course, the broader applicability of that would be to look at other forms of heart failure.
Okay. We're also involved with the Children's Medical Research Institute at Westmead, and that they've, as you probably know, and you alluded to some of that in Julia's presentation in relation to reforming adult stem cells to their more fetal stage. And I was just interested whether there's an avenue, Julia, for further sort of development of that research.
Sure, like if you're specifically interested, there are ways to make what we called patient-specific stem cells. So if we know, for example, a patient has a specific type of gene which is very interested, or in the future we would like to go into more precise targeted design therapy for patients, we can take the blood from those patients and make the unique patient line for either hypertrophic cardiomyopathy or for any type of pretty much disease. It's a potential to do this and make those models of those mini hearts specific to hypertrophic cardiomyopathy.
Okay. Thanks. Well, that's very interesting. Continue the good
Thanks.
work, folks.
Thanks, Peter. So I think that might be it. I think everyone's got to get back to patients and research, etc. So I just wanted to thank you all for coming today, the ones that are online now, but also we had plenty of people who also had registered but couldn't make the day, and so we'll get a recording afterwards. So thank you to all of you and the people who will be watching in the future. Thank you for your support. We obviously couldn't do the work without you all, and we're very grateful. But have a great rest of the day, and we'll see you at the next webinar. And thanks to all the
Thank you.
presenters today too.
Bye-bye. Thank you.
Bye-bye.
Thank you.
Thank you.